Very good morning to all of you. It is my pleasure to introduce today's speaker, Dr. S. Venkata Mohan, Senior Scientist from Bioengineering and Environmental Center of our institution. Dr. Venkata Mohan has completed his BTech degree in Civil Engineering in the year 1991 from SV University, Tirupati. From the same university, he has obtained his MTech and PhD degrees in Environmental Engineering during the years 1993 and 1998. Coming to his professional experience, he joined in BEC division in IICT in the year 1998 as Scientist B, and currently he is working as Senior Scientist. His area of interest includes environmental biotechnology and bioenergy and solving wastewater treatment and other issues. Dr. Venkata Mohan is a recipient of Humboldt Fellowship, and he did his postdoctorate at Institute of Water Quality Control Waste Management, Munich University, Germany during the years 2001 to 2002. He was also worked as a visiting professor at Research Center for Environmental Quality Management, Kyoto University, Japan, during the period 2005 to September 2005 to December 2005. Apart from his research activities, he is also serving as editor for the Journal of Energy, and also he is serving as member in several editorial advisory boards of several reputed journals. He has also served as member in Board of Studies in Environmental Sciences at SV University, Tirupati, and also at Department of Biotechnology, KL University, Guntur. Also, he has been serving as a member, Board of Governors of Biotechnology Research Society of India. Coming to awards and achievements, there is a big list. He has received several awards to his credit. He has received National Bioscience Award for the year 2012 given by Department of Biotechnology, India. He has also received Scopus Young Research Award in Sustainable Development for the year 2010. This is given by United Nations University together with Elsevier. It is worth to mention here that he is the first Indian selected for this award, which is given annually to young researchers. He is also a recipient of NACI Scopus Young Scientist Award in Earth and Environmental Sciences for the year 2010, given by National Academy of Sciences. In addition, he has also received Outstanding Reviewer Award for 4 consecutive years from 2009 to 2012 from Bioresource Technology Journal. Apart from this, he has received several Best Paper Awards. Coming to his research publications, he has published about more than 200 research articles, including 6 review articles in several reputed engineering journals with high impact factors. He has also published about 160 conference proceedings. He has also contributed to about 24 book chapters, and he got about 5 patents to his credit. He has also guided about 16 PhD students and more than 100 BTech and MTech students for their thesis. And currently there are 7 PhD students working with him. And one important thing that we can consider from his work is he is one of the most cited researcher with high citations, about 4,200, and also with a high H index about 37. So with this all, we can say that he is one of the most potential candidate for the future Bhatnagar Award. So with this small introduction, I invite Dr. Venkata Mohan to kindly deliver his lecture. Thank you.
Very good morning to all. Thank you very much, Sumana, for giving nice introduction. First, I would like to thank IICT lecturers seminars committee for giving me this opportunity, particularly Dr. Nahar Shastri, Dr. Rajendra Reddy, who encouraged me to do this presentation. With this presentation, I would like to share my work, which we are doing for past 15 years in IICT, extracting energy from waste by regulating the microbial metabolism. Coming to my academics, already Dr. Sumana told about it. I am basically civil engineer, specialized in environmental engineering, and I have done my PhD also from environmental engineering as specialization from Sri Venkata University, Tirupati, and joined IICT in 1998. And worked as a Humboldt Fellow at Technical University of Munich, and then as a visiting professor in Kuwait University, Japan. Coming to brief research profile starting from my career, initially I started working in physicochemical treatment methods using adsorption. I started my career in 1994 during my MTech dissertation work, research career. Subsequently, after coming to IICT, I was involved in various biological methods for treatment of wastewater, and I have also worked in various pilot plants and full plants. And subsequently, after moving to Germany, I was involved in advanced wastewater treatment methodologies such as periodic-based continuous batch mode operation. And since 2002, I reoriented my research in biological wastewater treatment towards energy generation, towards value addition. Apart from basic research, I am also involved in various applied research activities such as industrial and consultancy projects, and I have done some societal based research also, which I will present in the next slides. During my PhD, I was working in adsorption as a treatment technique. For this, I have used about 20 synthetic dyes related to various application and chemical classes, and tried to develop some low-cost treatment adsorbents for treatment of these dyes. And afterwards, coming to IICT, I also continued this work, but with biological treatment methods. In Germany, I have studied gene transfer mechanism in sequential batch reactor. Sequential mode operation is advanced treatment technique which was developed by my mentor in Technical University of Munich, Professor Wilderer, who is a Stockholm Prize winner. And there we tried to evaluate the horizontal heat transfer. Like we have used genetically modified Pseudomonas butera encoded with toll plasmid and tried to study it in a 32-liter sequential batch reactor. This horizontal transfer is the main mechanism for degradation of toxic pollutants. And we have used confocal scanning laser microscope as a technique along with fish. In Japan, I have opportunity to work in bioremediation, soil bioremediation, specific to coal-based hydrocarbons, where we try to use a specific reactor. In the first phase of reactor, we use surfactants to dissolve the pollutants, and subsequently in aerobic system, the dissolved pollutants were removed. And this system was again upscaled, and at present it's operating in the industry. Coming to focus of this presentation, at present my research, I would like to say our main focus is to evaluate potential of wastewater to generate energy or value addition by biological means, along with simultaneous remediation by keeping biorefractory as an integral part. Earlier perspective with respect to waste is different and energy is different, but at present people are looking waste as a valuable material and trying to generate energy from soil remediation, from biological treatment methods. This will give dual benefits like waste remediation as well as energy generation, and have very low carbon footprints. And moreover, it doesn't have any issue with the food to fuel issue, which is a big issue, and biofuels are considered. So our main perspective, our main goal is to evaluate the energy generation. My complete research reorients towards this one. Before starting a presentation, I will briefly explain what are the different biological routes we are using for generating of energy. When we take wastewater depends on the origin it is coming. It contains a lot of pollutants from organic to inorganic, and it may be simple organic molecules to aromatics, complex structures. And it again depends on the origin of wastewater. anyhow wastewater has to be treated, and there are basically biological treatment methods which are environmentally sustainable. Basically, there are two types of method, aerobic and anaerobic. After glycolysis, pyruvate will be formed in the biological step, and the pyruvate, when it goes to aerobic process in presence of oxygen as terminal electron acceptor, it will be converted into CO2, H2O, new cells through TCA cycle. But we are interested in anaerobic process. Where anaerobic process, the mature process nitrogenesis, where hydrogen acts as a terminal electron acceptor in producing methane. But again, methane is a greenhouse gas, and combustion generates CO2. So alternatively, we are trying to develop a process to generate hydrogen directly, which is a clean fuel, and combustion it will only produce water. So through acidogenic process, using proton as terminal electron acceptor, we are trying to regulate anaerobic metabolism to produce hydrogen. And similarly, we are also trying to generate directly bioelectricity in a defined fuel cell by using oxygen as a terminal electron acceptor at cathode by bioelectrogenesis. And we are trying to use anoxic microenvironment to generate bioplastics in the form of PHA using VFA generated from anaerobic reactor. And for past 5 years, we are also working on microalgae cultivation in order to treat as well as to produce bioenergy in the form of biodiesels. And I want to, from this slide, I want to stress that the anaerobic fermentation is a very versatile anaerobic metabolic function, which we can use for multi-value addition products like lactic acid, lipid, plastics, hydrogen, butanol, PHV, ethanol, like that. We can regulate the metabolism as and required, depends on output. So it's very exciting and versatile platform. And because we are using wastewater, when we see the wastewater, suppose we take distillery wastewater, generally distillery wastewater produce about 350 kiloliters per day. Single unit of distillery unit can produce about 350 kiloliters per day of effluent. This accounts for about 1 kiloliter alcohol produce 120 kiloliters of wastewater, 120 times the production thing. And about when we use hydrogenesis process, we can generate about 550 kg of hydrogen per day, which accounts for about 1.5 lakhs of additional revenue. Similarly, when we use microbial fuel cell with 20% efficiency, we can generate about 140 milliwatts per hour of energy, which again gives a revenue of 1.5 lakhs approximately. This shows the potential of waste which can generate energy, useful energy, apart from soil degradation. And based on this, the future, present the trend of treatment was only treatment, but in future it may shift to energy generation along with wastewater treatment, where we can replace the anaerobic reactors with either microbial fuel cells or anaerobic acidogenic reactors or bioprocessing systems, so that we can generate additional revenue apart from ETP operation. And based on this background, I would like to present our work in these 5 aspects. The first one is biohydrogen, bioelectrogenesis, biocatalysis, anoxidant photosynthesis, where we try to use wastewater for all the process and treated wastewater along with the generation of some additional revenue in the form of energy or a form of value-added products. First, I will start with biohydrogenesis. Biohydrogenesis is biohydrogen can be produced basically by 2 diverse methodologies, thermochemical and biological, but I am interested in dark fermentation, which is more robust and it can take wastewater as a substrate. Alternatively, there is a photobiological process. Theoretically, photobiological process can produce higher hydrogen than the anaerobic process, but the only problem is the limitations. It cannot survive in wastewater environment, and the oxygen generating from the process will not be feasible for hydrogen production. So we are trying to integrate at the second stage to dark fermentation, and there is an enzymatic process where pure enzymes are used to generate hydrogen, but still it's in the lab scale. Alternatively, recently microbial electrolysis method is being studied, but this method use a little amount of potential to reduce the thermodynamic barrier to produce hydrogen as an end product. And coming to dark fermentation, it's very complementary to the nitrogenic process, which is normally used for methane production in all effluent treatment plants, where we try to regulate the process at acidogenic step so that to form hydrogen as a metabolic end product. For that, we have used various metabolic regulation methods, which I will briefly explain in the next few slides. Generally, when we take 1 kg of COD we represent as a pollutant load in wastewater. When we remove 1 kg of COD from wastewater, we will be able to generate about 468 liters of hydrogen. This is the theoretical yield. And initially, during starting of the experiments, we tried to evaluate the potential of various wastewater. Is it feasible to produce hydrogen by this methodology? And we have used diverse types of wastewaters and observed good hydrogen production efficiency. But hydrogen production efficiency varied based on the composition of wastewater, organic loading rate. Simple the substrate, more the hydrogen. Complex the substrate, less hydrogen. But along with hydrogen, we also got good amount of CO removal efficiency, ranging between 30% to 90%, very few cases. And these are the bioreactors which we have used for evaluating. These are the lab scale 2 liters capacity systems. And this is the pilot plant reactor, 32 liters capacity, 50 liter capacity semi-pilot plant system, photobiological reactors. All these reactors were fabricated in our workshop. And during our study with various wastewater, we observed certain inherent limitations during hydrogen production. These inherent limitations restrict the hydrogen production and going towards methane production. I will briefly explain a few points. Coming to mixed culture, because we are using wastewater, we have to invariably use a mixture culture. Mixed culture contains methanogenetic bacteria as co-substrate. So we have to eliminate the methanogenetic bacteria in order to make hydrogen as end product. Otherwise, the hydrogen produced by exogenic bacteria will be converted into methane. So this is the major problem with the methanogenetic bacteria. And second one is fermenter inhibitors. Along with hydrogen, there is generation of volatile fatty acids as a co-substrate. And these volatile fatty acids reduce system buffering, like it reduces pH. When system pH reduces less than 4, the exogenic bacteria cannot function, and so hydrogen production gets stopped. This is another major issue. Because we are eliminating methanogenetic bacteria, we are not taking the reaction up to methane formation, where complete mineralization happens. So still, wastewater contains a lot of organic load present in it, like about 50 to 30% of COD is still present in wastewater. And what to do with this acid-rich effluent, because it's embedded with the volatile fatty acids and organic load. When we take wastewater as a basis, we have to treat up to regulatory requirement. This is one of the major issue. And along with the composition of wastewater and the carbon loading rates again influence the process. So initially we try to tackle with biocatalysts, like we are using mixed culture, we try to eliminate the methanogenetic bacteria. For that we have selectively enriched estrogenic bacteria by using some pre-treatment methods. We have used physical and chemical method. Suppose when we take acid soil treatment, we try to regulate the pH at less than 4 by parent inoculum, keeping for 12 hours, so that we can enhance the estrogenic bacteria, which is again a spore family bacteria. Likewise, each and every method has a specific function in enriching the either enriching estrogenic bacteria or eliminating the methanogenetic bacteria. Likewise, when chemical treatment is considered, BESA, it directly influences the enzyme which is involved for methane production. So we observed, we evaluated all these methods and observed all the methods for higher hydrogen production efficiency compared, several fold higher compared to the control operation. And this methodology was the basis for bioreactor operation in hydrogen production, which we published in 2008, which got very rapid citations, and this formed the basis for next our studies. With this encouragement, we have operated these experiments in series of bioreactors, with each system having specific pre-treatment method. Almost we operated 1 year, and among the operated methods, we observed heat shock and chemical BESA method are robust and sustained for longer hydrogen production compared to other pre-treatment methods. And during this phase, we also evaluated microbial diversity by PCR-BCG method and observed that selective enrichment of firmicutes was observed in all the pre-treatment methods. Like suppose this is the firmicutes compared to the untreated culture. Enrichment of firmicutes, firmicute phylum was the main responsible for hydrogen production, like Clostridia and Bacillus. And this shows the selective enrichment also based on biocatalyst. And next to biocatalyst selection, we try to optimize some of the critical factors which influence the biochemistry of the bacteria. Like we try to initially optimize pH, that we try to use 3 pH, 6, 7 and 8. And we observed 6 showed higher hydrogen production, whereas 7 showed towards methane production, because at 6, acidogenic bacteria cannot survive less than 6.5. So all our reactors we operated at pH 6, and we try to regulate, optimize the retention time. Lesser the retention time, more hydrogen production. Otherwise, when you keep it for longer times, the hydrogen produced in the headspace of reactor, it gets converted into methane. So we are getting maximum hydrogen production at less than 24 hours of retention time, where 12 hours shows the maximum yields. And similarly, batch mode and continuous mode operation, we observed batch mode systems are more amenable for hydrogen, whereas continuous mode are towards methane. And in this process, we also evaluated some trace metals which directly influence the hydrogen production. Suppose iron has direct influence on iron-iron hydrogenase enzyme, which is responsible for the hydrogen production in bacterial cell. So we selected some trace metals and optimized the trace concentration, which showed a significant increment in hydrogen production when trace metals are added along with wastewater treatment. Similarly, we optimize the biofilm configuration, where we get good hydrogen production in biofilm system rather than suspended growth operation, which is specific to methane production. And during operation with wastewater, the organic loading rate influenced significantly, because almost no single wastewater contains specific COD loading rate. Like when we take distillery wastewater, it contains about 150,000 mg of COD. When we take sewage, it only contains about 300 mg of COD per liter. So we want to study the optimum load which can take the acidogenic reactor to produce hydrogen. And we have taken food waste and operated a pilot plant system with 6 grams COD per liter to 50 grams of COD per liter. And with increment in organic loading rate, we observed significant increment in hydrogen production rates also. But after exceeding 50 grams of COD per liter, the hydrogen production dropped significantly. That is because the pH of the system dropped below 3, below 4. And this is because of formation of volatile fatty acids at higher concentrations, because COD loading rate was high. So the system buffering capacity got reduced and pH reduced, so lower the hydrogen production was observed. So we try to use some of the methods to regulate the system buffering capacity so that we can enhance some amount of hydrogen production. For that, we use 3 techniques. One is biogas reuse. Along with hydrogen, 50% of the biogas composition consists of CO2. We try to use CO2 to sparge into the bioreactor in order to enhance the system buffering capacity. By way of this, we are getting about 53% improvement in hydrogen production and 30% improvement in wastewater treatment. Similarly, we try to study the function role of feed replacement from influence of inorganic ions. And carbonate showed significant high performance with respect to enhancing hydrogen production, while phosphate ions showed good buffering capacity. During the process, we also tried to evaluate microbial diversity in different reactors, which was operated for almost 5 years using PCR and BCG technique with different conditions and different wastewaters. And interestingly, we observed all the systems enriched with firmicutes, which are hydrogen producers. And moreover, we try to isolate 20 potential strains from the reactors, and at present we are trying to use in the bioreactors for bioaugmentation purpose. And the final thing is how to utilize the acid-rich effluents, which is coming from hydrogen reactor. For that, we have adopted anoxic conditions, where the effluent coming from hydrogen reactor has high concentrations of volatile fatty acids in the form of acetate, butyric and propionate. We try to use it, we try to load it into anoxic reactor with facultative bacteria and created oxygen stress and nutrient stress condition, where instead of going to PCA cycle, it converts to PHB synthesis and we can generate bioplastics. By way of this, we are able to get about 50% of bioplastic in form of polyhydroxyalkanate, along with 80% of COD reductions. With this success, we are extending this study towards bioplastic production by using various wastewater by two-phase mechanism. And similarly, we also use microbial electrolysis by inducing small energy. The effluent, we can again generate about 20% of higher hydrogen production. But the major problem here is the power output, which is not feasible for application in full scales. And similarly, when we reduce the complexity of wastewater, we can generate additional hydrogen. That is the basis of this study, where we put a reactor with some electrodes, and so that because of electrochemical mechanism, the complexity of wastewater gets reduced. So afterwards, the outlet coming from the reactor, we feed into acidogenic reactor. By way of this, we are getting significant increment in hydrogen production along with COD efficiency. And recently, 2 years back, we got a project from MNRE where we have to upscale the process to 10 meter cube capacity. For that, we operated a semi-pilot plant system, hybrid configuration with biofilm and suspended, and evaluated to get design data for almost 2 years with different operating conditions, reactor configurations, and wastewaters. And based on that, is the photo video, but it's not functioning here. And based on that output, we integrated, because we are getting about 40% of theoretical conversion efficiency. The effluent we have integrated in the second reactor, and we increase the system buffering to pH 7. By way of this, we are getting about 50% of the theoretically hydrogen production theoretically. This we have upscaled to 10 meter cube, 10,000 kiloliters per day with 30 grams of COD per liters per operator, 30 COD per liter, and this is giving about 50% conversion efficiency. These are the figures where hydrogen and COD produced at different stages and going to finally ASP plant, mass material balance. And we try to evaluate the production cost, and we observed that when increment in wastewater treatment capacity is there, we can significantly reduce the potential cost. Similar to hydrogen production rate also, when we increase the wastewater treatment capacity, the rate of hydrogen production rate also get increased. And based on this experience, we developed a rapid protocol to evaluate hydrogen production. This is the 5 days methodology. So we call it as biohydrogen production potential. When we take any wastewater to the laboratory, within 5 days we can tell is it possible to produce hydrogen or not, with some conditions for upscale. The two-phase methodology. We can operate this for 5 days. This was published in International Journal of Hydrogen Energy recently. And in continuation to hydrogen, we are also working on microbial fuel cells. The microbial fuel cells are biologically catalyzed fuel cells. And the difference between hydrogen production here and microbial fuel cell is we are trying to introduce anode in the anaerobic acidogenic reactor where hydrogen is produced and try to extract energy directly instead of getting biogas and separating it and then sending to fuel cell. Here we can directly extract energy. Like when we give wastewater, through anaerobic metabolism, we get proton and electrons. And these protons pass through proton exchange membrane and enter cathode. This electron will attract towards anode and pass through circuit and go to the cathode chamber. In presence of oxygen, it gets reduced in presence of proton. So that cell EMF will be formed so that we can get energy in the form of energy. When we take wastewater point of view, 1 kg of COD removal, we can generate about 170 watts of energy. This is the theoretical figures. This is the close look of anode, cathode, and bacterial cell. Bacterial cell generate protons through cell membrane, and similarly electrons. Electron goes to anode, proton passes through proton exchange membrane to cathode, and resulting into cell EMF, by which we are able to generate power. As I already mentioned, these microbial fuel cells are biocatalyzed electrochemical systems, very similar to fuel cell, but the only difference is in fuel cells we are using chemical catalyst, but instead of that we are using bacteria as biocatalyst and trying to use the bacterial metabolism to catalyze the electrochemical reaction. And basically depends on the function, these biocatalyzed electrochemical systems can be segregated into four as biomicrobial fuel cells for generation of power, bioelectrochemical systems for generation of some value-added products like people are using it to generate ethanol by taking CO2 from atmospheric air, and microbial electrolytic cell where a little amount of potential is being given to generate value-added products, and bioelectrochemical treatment system where treatment was the main emphasis rather than the power generation. We are working on these two aspects which I will briefly present in next slides. MFC as power generator, similar to hydrogen production, initially we try to evaluate potential of MFC system to generate power by using varied varieties of wastewater from complex chemical wastewater to simple dairy wastewater, and all the systems showed a good power generation potential and interestingly more CO removal efficiency. Wastewater treatment efficiency was rather high compared to the biohydrogen process. In some cases, we observed about 90% of the CO removal efficiency. And similar to any biological system, we have to optimize the system to get maximum output. For that, we try to take some of the critical aspects which influence fuel cell performance. The first one is naturally pH, which directly have redox conditions directly have influence on biocatalyst activity. Here also we studied 6, 7 and 8, and 6 showed higher electrogenic activity with respect to proton release rather than pH 7 it is formed as either methane or hydrogen. And the second one we try to optimize the distance between electrodes, that's anode and cathode. Lower the distance, effective the electrogenic activity. And the next one is because we are using biocatalyst, the compactability of biocatalyst with anode is an important thing which depends the success of fuel cells. So we try to use some graphite along with low-cost metal electrodes and observe that graphite showed good electrogenic activity compared to metal-based anodes, along with good compactability for biocatalysts also. So we try to design all our fuel cells with graphite, and we also study the role of biofilm growth on anode, and we try to improve anode surface. Higher the anode surface, more electron will be attracted. So we use some nanotubes like multi-wall carbon nanotubes, carbon nanofoil, and after integrating the nanomaterials on surface of anode, they are getting higher electrogenic activity compared to plain graphite electrode. And similarly, one of the important component in microbial fuel cell is membrane, which is an expensive thing. So in order to make it more economically feasible, we try to develop some plant-based cellulose material, and we also use a fine sand, sediment sand and glass wool as a proton exchange membrane, and we got reasonably good results. Economic point of view, that results are very fine. And at present, we eliminated the proton exchange membrane, and we are trying to produce power. And another important aspect is metabolic function in anode and cathode. Because in anode, biocatalyst will be there. The metabolic activity and biocatalyst nature influence the electrogenic activity. This is the first of the study. We try to evaluate aerobic, anoxic and anaerobic mechanism by inoculating respective bacteria. And interestingly, we observed that the aerobic system showed higher electrogenic activity compared to anoxic